We have previously explored the descriptive epidemiology study design, which is cross-sectional studies, case series, and case studies. Now, let's move to analytic epidemiology studies. The first study design in analytic epidemiology study is case control study. The purpose of case control study is to establish association between exposure to risk factors and disease. So in case control studies, members of the population with the disease are selected into the study at the outset and risk factor information is collected retrospectively. This group is known as cases. A second group of individuals who do not have the disease is also included in the study and they are known as control. Case control studies are often used in the study of rare disease or preliminary study, where little is known about the association between risk factors and disease of interest. If you look at this figure, case control study is the red one, is when you collect a data from the present time, but you are interested to know what have happened in the past retrospectively. So in case control design, the study population is divided into two groups, which is disease or cases and without the disease who are control. And from each group of cases and control, the factor is assessed, the exposure is assessed whether the factor is present or absent, present or absent. But these factors are from the past. The state of the disease is the present. Therefore, case control study looks information retrospectively. Case control studies are prone to bias and confounding. To minimize bias, care must be taken in the selection of cases and control, in establishing definitions of disease and the risk factors, and finally, care must be taken in ensuring that there are no confounding association between the detection of disease and risk factors exposure. Care must be taken when choosing cases for this case control study. It is important to distinguish between stages or subtypes of disease and to define a measure of health status. Example, when studying physical activity, it is important to define what is meant by physical activity in terms of types, nature, and level. It is also important to establish whether interest is in incident cases, that is, subjects entered into study on detection of disease, or we call them as newly diagnosed, or prevalent cases, those who have been diagnosed as having the disease prior to the study. It is important to take note that views, behavior, and reports of exposure to risk factor amongst incident cases and prevalent cases will tend to differ. Those diagnosed previously are likely to be more informed about the disease and may have altered their behaviors and attitude. Incident case design is preferred as it reduces recall bias and overrepresentation of cases with long-standing base. There are also few concerns when we choose control. Controls should come from the same population at risk of disease. Control should not have the disease, means that they are healthy, and they should be representative of the target population. Selecting controls often proves harder than cases and requires great care in the prevention of bias. A sampling frame of hospital patients is often used to select controls. However, risk factors such as diet and smoking are commonly linked to many diseases. Selecting controls from hospital patients' sampling frame might therefore overestimate population exposure. underestimation of association between disease and exposure, and to overcome this, we must use more than one control group. Multiple controls can be used for each case, and this will give the study greater power. And it is particularly important where the number of cases is small due to disease being rare. Another aspect on reducing bias in case control study is looking into the exposure to risk factors and matching. Exposure measurements are reliant on memory when the person is interviewed retrospectively and or when we refer to medical records. Exposure estimates are vulnerable to recall bias when those with disease are more likely to remember exposure than those without. Exposure estimates are vulnerable to interview or measurement bias. Interviewer interviews or reports findings systematically differently between cases and control and confounding factors. And this can be overcome by including blinding in the design so that they do not know who is a case and who is a control at the time of interview. In addition, confounding factors must be identified prior to the start of study. Individuals match to controls where it is thought that other factors aside from those risk factors of interest might contribute to the development of disease. Example, their age, sex, ethnicity. The factor upon which cases and control are matched cannot be studied as a risk factor. Alternative method of overcoming confounding is to collect relevant information on them and adjust them statistically. Matching is used in case control study to permit allowance for confounders which are complex or difficult to define. Example, by comparing within identical twin case or control pairs, it is possible to allow for ill-defined genetic confounders. Matching is also used to make allowance for confounders statistically more efficient. Efficient analysis requires that there be a similar ratio of cases to control at each level of exposure to the confounding variable. Matching is also used to reduce biases in the ascertainment of exposure. Example, data collected from disease cases should be matched to disease control as information is collected from their family members. In terms of data analysis, case control study measure the odds of exposure based on disease, compared to cohort study which measure relative risk of disease based on exposure. Odds ratio have two components, that is, the odds of exposure for cases and the odds of exposure for controls. This slide shows how to calculate odds ratio. We have learned on how to calculate odds ratio in previous lectures. Therefore, to summarize, the estimate odds ratio is when odds is equal to AD over BC. We are still in analysis of data for case control study. Assessment of whether an observed association is likely to be directly causal and not the results of unrecognized confounding depends on, first, the size of the relative risk, where higher relative risks are less likely to be explained by unknown confounders. Second, the presence of a dose-response relation, where the observation of a higher risk in subjects with greater exposure to the risk factor favors a direct causal relationship. And third, the existence of plausible biological mechanism which might explain a causal relationship. There are three main advantages of case control studies. The first one is it's quick to be conducted. Second, it's cheap. And third, it's particularly suited to the study of rare diseases as the diseases are selected at the outset of the study. There are five disadvantages of case control studies. The first one is difficulties in overcoming potential bias and confounding. Second disadvantages is the successful selection of both cases and control who are representative of their respective population is often difficult. The third disadvantage is an ability to infer causality and no information on the chronology of disease and exposure because of the nature of study which is retrospective. Fourth, disadvantages is inefficient in studying risk factors which are rare. And finally, studies are often not population based, therefore, it is impossible to calculate incidence of disease.